Friends, welcome you all on the behalf of Rahul Anand Biology Tutorials. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand Biology Tutorials. As well as you are very clear about that, means today we are going to study about what? So today we are going to study means what are actually the molecular bio here means in terms of the molecular basis of inheritance. So these are the particular name of the topic as well as here in terms of the chapter. After that when we are dealing about these means what are actually the topic variants. So today we are going to study about these means in terms of what? In terms what are actually the genetic code. So genetic code are what? So the genetic code are the phase of the mRNA or DNA, which is playing important role to determine what? Which is playing an important role to determine the sequence of the amino acid, and particularly after that what will be happening? So it is often playing important role to determine the here in terms of the sequence of the protein. Now, so when we are talking about these cases in terms of the DNA, so you are very clear about that, remember? So the genetic material are the DNA, means the deoxyribonucleic acid, as well as we are having the RNA, means the ribonucleic acid. So the RNA is not the genetic material, but when we are talking in the case of the certain of the viruses, so it will be considered under what cases? So it will be considered under the case of the genetic material such as the retroviruses as well as the reoviruses. Now, so often when we are talking about the case means what are actually the amino acids. So amino acid are what? So the amino acid are the micro molecules or the particles with reference to the protein. Remember? As well as when we are talking about the protein, so protein will be what? So when many of the amino acid will literally combine, it result in the formation of what? So when many of the amino acid will literally combine, it result in the formation of the protein. Now, so when we are talking about these cases about the protein, so it is oftenly being further divided in terms of the broad categories, such as we are in the fibrous protein, then the globular protein. So we'll be understanding about that later. Now, so when we are talking about these courses in terms of what? In terms of the genetic code. So primarily in this case it will be initiated with reference to what? So it will be initiated with reference to the DNA. After that in terms of the RNA, then in terms of what? Then in terms of the mRNA, and finally in terms of what? And finally in terms of the protein synthesis mechanism will be employed in this case of the genetic code. As well as when we are talking about these cases, means what does these mean in terms of the genetic sequence playing an important role. So these genetic sequence are playing important role in order for what? In order for determining the phase with reference to what? With reference in the case of here in terms of the mRNA sequence, then in terms of what? Then it is often playing important role with reference in the case of the nitrogenous bases. After that what will be happening? So after that in terms of the protein synthesis molecule as well as the mechanism will be literally employed as well as taking place. As well as when we are talking about these cases, means what are actually the genetic code. So in this case particularly we are having the two important as well as the vital process. So these are literally known as what? So these are known as the transcription as well as here in terms of the replication. Now, so when we are talking about these cases, means what are actually the transcription as well as here, tell me in terms of the here in terms of the replication. So in the case of the replication what will be happening? So in the case of the replication, the DNA will be copied with reference in the case of the DNA in terms of the deoxyribonucleic acid only. But when we are talking about the case in terms of the transcription, so in this case what will be happening? So in this case, tell me here in terms of the what? Tell me in terms of the DNA, means the deoxyribonucleic acid will be converted as well as from the DNA. So from DNA the mRNA will be literally formed. So these are literally coming under what case? So these are literally coming under the case of here in terms of the transcription. As well as when we are talking about these cases, means in terms of what? As well as when we are talking about these cases, in terms of the transcription as well as the translation. So these are literally showing the many types of the here in terms of the complexities as well as the similarities as well as the various kinds of the variants in it. As well as when we are talking about the another cases in terms of what? As well as when we are talking about the another cases in terms of these, tell me in order, means what are actually the mRNA as well as these mean in order in terms of the RNA. So in this case what will be happening, or either you could say that in the case of here in terms of the genetic code process, so what will be happening? One nucleic acid will be what done? So one nucleic acid will be copied in another in terms of the another nucleic acid. So this is the process somewhat coming under the case of the genetic code. After that, when we are talking about the another features, so we are having the different types of the vital stages in it, such as we are having what? Such as we are having the single codon model, as well as the double codon model, as well as the triplet codon model. As well as when we are talking about these cases in terms of the genetic code process, so primarily it will be initiated with reference to what? So primarily it will be initiated with reference to the deoxyribonucleic acid. Then what? Then in terms of the mRNA. Then after that we are having the four vital codes in terms when we are talking about the case of what? In terms of the nitrogenous bases, which are literally coming under what case? Which are literally coming under your here in terms of the RNA case. So yeah, so these are what? So these are the adenine, then the uracil, then the here main term of the guanine, then what, the main term of the cytosine. Of course, after that, when we are talking about the different codes as well as the basic here main term of the defect of these main term of the code, and when we are talking about these cases, mean in order in term of what, mean in order how these particularly genetic sequence process literally work. So primarily it will be initiated with reference to your deoxyribose nucleic acid. After that, what will be happening? So after that, the mRNA will be formed, and from this mRNA we understand that we are having the 4 different types of the nitrogenous bases. So these are what? So these are the adenine, then the uracil, then what? Then the cytosine, often the guanine. Of course, such as in terms of the AUCG, of course. As well as when we are talking about these cases, so these are what? So these will literally combine in order to form the specific type of the protein. As well as when we are talking about the case of the protein, so we are having around how much? So here we are having around 20 different types of the amino acid in terms of the protein. Just like we are having what? Just like in the case of the English alphabet letters, we are having what? So we are having the 26 of the alphabets letter, etc. When we are talking about these cases, tell me in terms of here we talk the 20 different amino acids. So these are what? So these are nothing else but the adenine, guanine, cytosine, uracil. Often you will be having the valine as well as the glutamic, etc. Glutamate, etc. So we will be understanding about these cases later. Now, so we are talking about the basic defect as well as the problem of these in order of the genetic code. So your basic problem as well as these in order in terms of the genetic code is what? So the basic problem as well as the defect of the genetic code is to find what? So your basic problem as well as the defect is in order in terms to find the nucleotide codon in terms of what? In terms of the nucleotide codon. So when we are talking about these cases, means what are actually the basic problem as well as the defect of genetic code will be what? So your basic problem and the defect of the genetic code will be this in order to find the here mate of the specific type of the nucleotide codon. Of course, as well as when we are talking about these cases, means what are actually the different types as well as the parts. Tell me in terms of the codon rule. So we are having the singlet codon rule there. Then in terms of the doublet codon rule as well as the triplet codon rule. So when we are talking about these cases, means what are actually the singlet codon rule. So it literally tells that what? So here main term the one codon will code what? So here one codon will literally code in terms of the one nitrogenous basis. Okay, fine. So when we are talking about these cases in terms of the singlet codon formula, as well as when we are talking about these cases in terms of the rule of the singlet codon. So it tells what? So it tells that what? So it tells that one codon will code one of the nitrogenous basis. As well as when we are talking about the formula, so these are bearing the formula of 4 to the power n. Literally, when we are talking about these cases, mean in terms of what? Mean in order in terms of 4 to the power n or either the 1. So it literally means what? So 4 into 1 is equal to only the 4. Mean of the 4 codon. So here we have just understood one codon will code for how much of the nitrogenous basis. So here mean of the one codon will literally code for one nitrogenous basis. Likewise, the 4 codon will code for 4 nitrogenous bases. Now,